Hi, everyone. I'm Kirsten Kenneberg-Mahalik, Downstream Marketing Manager here at Repligen. I'm so glad you could join us today for the third episode of Repligen's 2021 webinar series. In the first episode, we introduced a new affinity resin for the purification of protein-based COVID-19 vaccines. And in the second episode, we went upstream to the bioreactor and discussed an intensification device. You can access both of these presentations on Repligen's events page. So if that brief overview has piqued your interest, I would encourage you to check those out. In today's episode, we're back to chromatography. We are lucky to have 3 technical experts joining us to discuss how you can leverage technology solutions from Repligen, Tecan, and Synthase to design and deploy an easier, smarter, and faster workflow to accelerate process development. From Repligen, we have Tim Schroeder, Director of Product Management. From Tecan, we have Jana Langhoff, Product and Application Manager. And from Synthase, we have Dr. Arne Vandebroek, Senior Automation Engineer and Product Lead for miniaturized purification. Before I pass it over to Tim, there are just a couple housekeeping items that I have to address here. The first is that this webinar is being recorded. This allows us to post the webinar on our events page so that anyone who wasn't able to join us today can watch the webinar on demand starting next week. The second is that we have a live Q&A panel on the side of your screen. You are welcome to submit a question anytime during today's presentation, and our panel of experts will address those questions at the end of the talk. You'll also have the opportunity to upvote questions that you're particularly interested in, so be sure to keep an eye on that Q&A panel throughout today's presentation. And finally, we have 2 poll questions that we'd like you to participate in. The first question asks about your experience with miniaturized chromatography columns, such as our Opus Robo columns. And this question just helps us get a sense for who's here today. So if you could pop into the side panel, make sure you're in the tab for poll questions and not the tab for Q&A, and answer that first question. We'd really appreciate it. When you're done with the poll question, make sure you switch back to the Q&A tab so you can see the live feed of questions as they're being submitted. At the end of the presentation, we'll post a second poll question, so stay tuned for that. I hope you're all as excited as I am to learn more about how you can enhance your RoboColumn workflow with Tecan and Synthase. So with introductions made and housekeeping items out of the way, I will pass it over to Tim to get us started.
Thank you for your kind introduction, Kirsten, and also welcome to everybody in the audience today. It's actually my great pleasure to kick off the shared presentation between Repligen, Tecan, and Synthase, and I hope that we are able to demonstrate today how you can benefit from an end-to-end solution which has been developed for the operation of Opus RoboColumns. But before we go into the details, I would like to take the time to briefly introduce you to Repligen's innovative solutions for bioprocessing. And this is quite an interesting story to tell, as Repligen has been primarily known as a leading supplier of protein A ligands into biopurification workflows since the mid-80s. But since that time, and even if Repligen remains the world-leading supplier of protein A ligands, our footprint in bioprocessing has been tremendously changed. We meanwhile understand ourselves as a full technology provider of innovative solutions into all steps of modern bioprocessing, covering upstream processing, downstream processing, as well as analytical workflows. Besides the XCell ATF systems based on hollow fiber technology and used for the concentration of cells during upstream processing, and TangenX flat sheet cassettes, Opus prepacked chromatography columns have been broadly adapted by global industry over the last decade. Opus prepacked chromatography columns have been designed as an open platform, which can be user-specified or rather prepacked to any diameter, any bed height with any resin. The fully integrated portfolio gives you an unmatched diversity of available formats, each format targeting a different stage during your downstream operational process. And this allows you as a customer to seamless and reliable scale up from early process development through process validation into GMP manufacturing. However, today's focus is on Opus RoboColumns, which have been developed for the operations with Tecan liquid handling robotic workstation, and which are meanwhile broadly adapted by global industry for downstream process development, so-called screening, but also for sample preparation prior to subsequent analytics, supporting drug discovery and other upstream workflows. Prior to the availability of RoboColumns, downstream process development has been traditionally carried out sequentially in a single column mode of operation on LC systems like Actor. And this has been leading to extensive and significantly extended process development timelines. And also in more detail, the time required to plan, prepare, and execute your experiments and the following data and analysis are entailing the risk that you are missing the most relevant process parameters. And even if this has been tremendously changed, the situation has been tremendously changed over the last couple of years, and design of experiment has been a common approach to plan your experiments, we notice that the 3-unit operations are still more or less disconnected in many cases, and that our customers are spending a lot of time to integrate all these units and have a robust workflow. And therefore, the goal of our cooperation between Tecan, Repligen, and Synthese has been to go beyond that and to be prepared for modern, new, future process development needs by creating an end-to-end solution which allows you as a customer to do a robust DOE planning, to carry out your experiments in a timely manner, and to have straightforward data assessment. And this is our solution, which is actually the result of a great and grown relationship between Repligen, Tecan, and Synthese, where each industry partner contributes with a modern, state-of-the-art, innovative technology addressing a different requirement in process development workflows, and finally, all together resulting in a fully integrated solution for you as a customer. On the one hand side, Repligen provides the RoboColumn platform, which enables parallel execution of your experiments. Tecan supplies world-class liquid handling robotics and workstations. And finally, Synthese developed and supplies Anta, a new software for experimental planning and data analysis. So let's have a closer look how a new integrated robotic workflow can look like with Opus RoboColumns. So typically starting with experimental planning, following a DOE approach, followed by material preparation, experimental execution, and data management. And this is driving you to a decision point where the decision has to be made if the process is already robust and good enough to further scale up, or if the process needs to be reviewed, readdressed, and a new DOE has to be carried out. And with that, and in order to demonstrate how the new Anta software can help you to streamline your experimental planning, I would like to hand over to Arne from Synthese.
Thank you, Tim. So I'm going to introduce Anta, which is a piece of modern cloud software that simplifies sophisticated experiments. And core to Anta is the drag and drop workflow creation interface, which you can see in the middle of the screen. So all this happens in your web browser from anywhere in the world. You can drag and drop elements that define your experiment. We have elements that define the stages of your chromatography experiment, where you can define which buffers you're going to use, where you can define how many CVs you want to use. You can even specify whether you want to collect blanks or not for data correction later on. You can also specify your samples, your buffers, and your resins. Once you are happy with that experiment, you can simulate that experiment. And that brings a key feature of ANTA. It allows you to preview your experiment, which you can see here on the right. What we see on the right is actually the preparation of a number of gradient buffers to be used in a gradient buffer experiment, all within the same modern software experience. Because ANTA parameterizes the entire workflow with these elements, it now becomes straightforward to import DOE design files. You can have DOE design files made in JMP or Design Expert and upload them into ANTA. ANTA will take these design files and translate them into machine instructions. You don't have to worry about aspirate and dispense cycles. ANTA does that for you. All you need to do is say, run one has these factors, and I'm going to set them at these values. Now I'm going to pass back on to Tim. This is the end of the planning part of ANTA, and I'll talk in a couple of minutes about ANTA's data management capabilities.
Thank you, Arne. So let's spend a few minutes on the second step of our new integrated workflow with RoboColumns, which is actually material preparation. Even if we are all aware that material preparation is not limited to column packing and includes other work such as buffer preparation, we can align that column packing is still a time-consuming task, which also requires skill set on the operator level. So the good news is this burden can be completely moved away from you by outsourcing all column packing activities to Repligen, in particular for Opus RoboColumns, which are required for the application we are discussing today. Opus RoboColumns are miniaturized real pre-packed chromatography columns, which can be arranged onto a 96 array format according to application requirements, and which enables you to operate 8 RoboColumns in parallel. Opus RoboColumns are available in different volume configurations, actually covering a volume range from 50 to 600 microliter. At the same time, Opus RoboColumns are available pre-packed with a huge diversity of more than 300 resins. And for more detailed information, please visit our webpage where you can download a complete comprehensive list of available resins in Opus RoboColumns. And with that, I would like to hand over to Jana from Tecan, who explains how RoboColumns can be operated with Tecan's liquid handling robotic workstations.
So thank you, Tim. Execution actually of the experiment is then becoming really easy with the integration of ANTA for TCHROM, because the experiment goes straight from laptop to lab. Thereby, the ANTA-generated instructions are automatically converted into liquid handling robot compatible commands using the Fluent Control or EvoWare API. So then the liquid handler goes straight ahead and carries out the design purification experiment using the Opus RoboColumns without any further intervention. The beauty of automation is really that you can get done more in less time, but at the same time maintain precision and accuracy. The Tecan TCHROM module has been developed together with Repligen specifically for the Opus RoboColumns and has been refined and improved over the last 13 years, so that we have now almost 300 customers worldwide relying on this technology and making Tecan's liquid handler the preferred instrumentation for this application. On the Fluent, we can aspirate and dispense liquid handling with 8 independent channels, including regulation of dispense speed, which then directly translate into residence time for each channel individual actually, and as low as 8 centimeters per hour. On the middle to large sized instrument, as seen here in the picture, we can combine column chromatography and plate-based process development with other downstream experiments to provide as much flexibility and use of lab space as possible. So whether you need a small dedicated workstation that just runs your RoboColumn experiment on a Fluent 480, a Freedom Evo 100, or a dedicated pre-configured application package, or if you need to automate an entire workflow to enhance productivity and eliminate all manual process steps, including buffer preparation or collection of fraction and absorbance data, we have you covered with almost unlimited options, but individual solution tailored to specifically your needs on the Fluent 1080 or Freedom Evo 200. With the integration of modules such as a laminar flow hood with HEPA filtration or an entire Class 2 enclosure, but also centrifuges, shaker, vacuum station, temperature-controlled deck space, incubators to increase incubation times, or increased plate storage, to just name a few. Because we are able to run 8 RoboColumns in parallel, we can reduce the purification processing time by a factor of 8. So an experiment that would usually last, when run sequentially, up to 25 hours can now be done fully automated and without further intervention in just about 8 hours. And with this, I would like to hand over to Arne, who will discuss the ANTA software integration with Tecan's liquid handlers.
Thank you, Jana. As you can see on this slide, both the Opus RoboColumns and the ANTA software is supported to work in conjunction with the Freedom Evo liquid handler. I'm very excited today to be able to announce that we're going to have support for ANTA using RoboColumns on the Fluent liquid handling system. That is coming mid-June. We're going to have another webinar talking about that. These purification experiments often require many software touchpoints and have fragmented data files. And this is one of the key issues that ANTA aims to resolve. You have the purification experiment on the left here. ANTA already helps to resolve the buffer preparation and to define the stages of your experiment. If you want to add a stage, that's very easy, very straightforward to do within ANTA software. But very often when you want to do analytics, you need to do some kind of sample preparation. Now that is also included within ANTA. So for example, if you want to determine yield, you are going to use a plate reader to determine the concentration of your elute fractions. But your concentration may be too high. So you may want to dilute your samples by a factor of 10, for example. Now this is very easily done using ANTA. You can specify dilute by a factor of 10, and ANTA will then multiply the resulting concentration by that original factor of 10. So because it knows that it has done these dilutions. Now, similarly, if you want to do normalization for HPLC analysis, that's also possible. So that sample preparation step is naturally included within the ANTA software. But the most exciting thing is that ANTA is able to combine a variety of analytics. Typically, you'll have, like I said, yield data coming from, or at least concentration data coming from a plate reader. You might have purity data coming from a lab chip device. You might have characterization data in terms of host cell protein from ELISA, or you might have aggregates or fragments from an HPLC study. That results often in 5 to 10 different CSV or Excel files. And you struggle to get that data back, align that data back with the original factors of your experiment. Now, ANTA does that automatically. When you can upload data from these various analytics, ANTA will automatically align it and augment it with metadata. So once an experiment is finished, ANTA really helps at eliminating the time-consuming and error-prone manual data handling that we often struggle with in the lab. So data from the plate reader that is integrated on the Tecan platform is automatically aligned with the liquid handling steps. And you're able to immediately, after the conclusion of your experiment, you will immediately see the raw data in terms of chromatograms that are path length corrected and also blank corrected if you've chosen to collect blanks. So you can see on the right here a table showing sort of the data alignment, and underneath you can see these preliminary chromatograms. On the bottom you see the cumulative data where we sum all the the properties, for example, of the elution step together, and we have a combined yield or purity. So ANTA allows you to also easily select samples for downstream analytics. So you can say all the samples above a certain concentration I want to send for HPLC. Here I wanted to present a case study that we did in our labs at Synthase headquarters in London. It's a case study that characterized transaminase purification using space-filled DOE design. The objective of the study was to maximize yield of recombinantly expressed transaminase while maintaining 80% purity. So we have both yield and purity. Yield is obtained with the plate reader. Purity was obtained using the lab chip electrophoresis. The table on the left shows the 9 factors that we studied. We had 6 different resins. We had 3 different pH levels for load, wash, elute buffers. We varied the load and elute flow rates. We changed the sample load. We changed the wash buffer salt concentration and also the wash volumes. So these are 9 factors with each different levels. So this is actually quite a massive parameter space. We extracted 48 runs out of there and send them to ANTA to calculate the instructions. We split up the experiment in 2 × 24 runs so they could be run over 2 days. Each of these runs took 6 hours, so that's like a really good working day with some preparation time. So it was 2 × 6 hours. ANTA was able to combine the data taken over these 2 distinct days into one. So that's a great example of ANTA's functionality. Now, we are at Synthase very enthusiastic about these sort of space-filled designs because they help to avoid cliff points. In traditional DOE designs, the corners of your design are very important. But with chromatography, sometimes it happens that you have a very limited product yield in a corner of your design space. That means you would lose these data points. That means your DOE modeling becomes quite cumbersome. In contrast, if you use the space-filled designs, every point is sort of less individual importance. And the whole point is when we set up these sort of complex space-filled designs, you need automation. You need Tecan's liquid handler. You need the miniaturized columns. Well, we are very enthusiastic about the combination here of the software that enables to translate these designs and get the data back, the automation, and the miniaturized columns, because now you need much less sample to do all these experiments. So you can actually study many more factors. What such a space-filled DOE design taught us was really to give a bird's eye view on the many factors that affect the purification process. We did see interaction effect in this study, as you can see on the plots on the lower left on the slide. So that really means that in a process like this, purification process like this, there are interaction effects. So you should study these parameters simultaneously. So you should be doing DOE to get a good view of the design space. When we did this in this study, what we learned is sort of out of these 6 resins, the top 2 that performed well. And we also learned that we needed to expand the design space. And that goes back to that arrow in the graph to go iterate through your design. A study of this magnitude is simply not feasible without miniaturization, robotics, and automation. Setting this up in the lab is really complex. Preparing buffers is tedious and error-prone. Keeping track of all the factors is a true nightmare if you have to do it manually. I will now handle it back to Tim for conclusion remarks.
Thank you, Arne. This case study does a very great job demonstrating the data analysis, which is part of our new integrated workflow with RoboColumns. So this is actually the last slide of today's presentation. And I would like to take the chance to briefly recap on the integrated workflow together with you. We covered experimental planning, following a DOE approach. We prepared the material. We executed the experiments with the help of robotics. And finally, we just completed the data assessment, which is leading us finally to the blue diamond in our workflow. And this blue diamond is actually standing for the decision-making point where you as a researcher needs to decide if the process is already robust and solid enough for further scale up, or needs to be readdressed by adjusting the DOE and following the complete integrated workflow again. So what we are really talking about here is recognizing this decision-making process and supporting it with more, better, and faster data by leveraging the key benefits of each of the three technology providers. Expertise in column packing from Repligen, fast and parallel execution of experiments with Tecan robots, and ease of use with Synthase modern software. All together is leading to an enhanced process development workflow with significantly reduced cost per experiment through saving sample, and finally resulting into faster time to market for you as a customer. On the other hand side, and if you decide to further scale up your process, you have access to Repligen's Opus pre-packed column portfolio, packed with any resin in a flexible range from 0.5 to 80 centimeter, which allows you to continue your downstream process development work rapidly with speed and reliable, as well as seamless scaling up into GMP manufacturing. At the end of today's shared presentation, I would like to take the chance to thank both Jana from Tecan and Arne from Synthase for their great contribution to this presentation, as well as everybody in the audience for your kind attention. With that, I would like to turn back to Kirsten, who is going to moderate the QA session.
That was great. Thank you, Tim, Jana, and Arne. I also want to thank each of you in the audience for participating in this episode of our 2021 webinar series. I'd like to ask once again that you pop into the side panel and answer our second poll question. So thank you in advance for doing that. Before we jump into the Q&A, I'd like to bring your attention again to the webinar that Arne mentioned. For anyone who's interested in learning more about Synthase's new capability with Tecan's Fluent Liquid Handling Workstation, I would encourage you to visit Synthase's website. You can find them at synthase.com. From there, you'll be able to register for their demo webinar, which is scheduled for Thursday, June 17th. Okay. Let's jump to some questions from our panelists. I'm going to start with the most popular question here today. At the top of our leaderboard, if you will, we have columns are not held straight in the holder plate, leading to tip damage. Is this being addressed by Repligen or Tecan? I'm going to hand that question over to Tim.
Yeah, thank you for this question. And this is a serious concern which we have noticed or which has been reported during the recent months by many customers. So we have been sitting together with Tecan and we have been brainstorming regarding these observations or this complaint. And the good news is that we have been coming to a long-term solution, which we call the RoboColumn downholder, and which would be a spare part for the already established T-Chrome module, which allows you to fix and hold the RoboColumns onto the array plate in position. And then there's no chance that they will be removed from the plate during operation.
All right. Thank you, Tim. The next question here. Does the Antha workflow handle issues with repeat injections onto the RoboColumn O-rings? The stop flow is a problem for my chromatography. So Arne, can you jump in with that about the Antha workflow and how it would handle repeat injections?
Yeah, so in principle, that's up to the user to decide whether you want to do repeat injections or not. But typically what we do is we multi-dispense into the columns if you want to collect separate fractions. So we would fill up to the diluter volume that you have available, whether that's 1 milliliter or 5 milliliter. That's all specified in the configuration. We would then fill up to that amount and then dispense using a multi-dispense sequence, so without ejecting the tips out of the columns into whatever fractions you've specified. So it's all really flexible, which allows you to sort of adjust it the way you want it to behave.
Great, thank you, Arne. And it looks like we have another one that's coming your way. Is the Antha preview for a generic deck, or is it specific to my TCAN deck? So can you talk a little bit about how that in silico preview is generated?
Yeah, that's actually a really good question. So when we deploy Antha at the customer's site, it comes actually with 2 pieces of software that run on the computer connected to the robot. One piece of software is called Get Configuration. So that piece of software actually extracts the configuration from your device, including the different deck work table layouts that you may have. And then when you run Antha, you can select which work table layout you want to plan against. And so that your exact work table layout will be mimicked in the preview. So the preview really is sort of, it's a view about the instructions that you will see on the robot. So let's say you say, I want to aspirate from, let's say, my troughs, and then have, let's say, an elution into the robocolumns. Those liquid handling actions of aspirate and dispense will be visualized as a transfer on the preview. So it really allows you to verify all the steps in your process. You can even select per chromatography stage what will happen, and it's very nice to see the overview of your experiment.
Let's stay on the Antha software. I know I was running sort of with popularity of upvotes here, but let's stay on this topic for just a little bit longer. Arne, can you comment, is the Antha software Part 11 compliant?
That's at the moment we are working towards that. So the answer is no. We are not officially compliant with CFR P11, but that's something that we're working on internally to provide in the near future.
Okay, and still, again, staying with the software for just another question here. Does the Antha pipetting handle dilution effects that are typical of a system liquid-filled pipetter? It looks like Daniel is saying my elution profile is compromised due to this.
So there's several ways to sort of mitigate the dilution effect. Is you could use large excess volumes. We did actually some studies in our own lab regarding to that. So excess volumes is the solution that we have preferred because it's good enough and simple. Another solution, which is actually better, is using partitioning volumes, where you would aspirate small amounts of liquids, maybe in 1 or 2 aspirations, before you aspirate the buffer or the sample that you want to load into the columns. We don't operate in that mode too much because it's a little bit more complex to provide. So far, our customers are happy with the excess volumes, but we could, if there would be customer requests to implement the partition volumes, that would not be too difficult for us. That's something that we have sort of on the back of our minds that we may want to implement should the excess volumes deemed not to be sufficient by some customers.
Okay, let's turn our attention to the columns for a little bit here. Tim, will these columns be made available for other systems, or are they system specific?
It's difficult to give a straightforward answer to this question. So in general, Opus Robo columns can be operated with any liquid handling robotic workstation, which allows positive liquid displacement technology, so controlling the flow rate. Anyway, we have been working with TCAN since the early development of those columns almost 15 years ago. And that's why the columns have been exclusively constructed to fit against TCAN stainless steel tips. Right, and since then, TCAN has been significantly invested into developing hardware as well as software solutions, and also the Synthase Corporation is finally part of that. So we really recommend to use a TCAN liquid handling robotic workstation in order to allow a robust and solid, sustainable workflow.
Thanks, Tim. Let's stay on the column and let's circle back to something we touched on earlier. We have a follow-up question here from Deirdre. When will the downholder be released? And will it be available to work with different column sizes, or will there be a downholder for each column size? So Tim, can you take that one as well?
I think we should move that to Jana, because the downholder will be actually a spare part from TCAN, but it should be launched very soon. But maybe I can comment on the column sizes. There are just two different outer column geometries, long and short robocolumns, and the downholder will fit both column geometries. But anyway, you are not able to mix both geometries in one workflow at the same time. So this doesn't matter.
Jana, do you want to chime in there? I want to make sure I give you the opportunity if there's anything you want to add.
Exactly. So like Tim said, we are working on this full steam and have been for a while, and they're about to launch. So I don't have a date yet, but I really hope that in the next month this will go through, and we will definitely notify everybody that we know that is using the robocolumns. And as Tim also said, they of course will be, so one column downholder will adjust to both of the sizes, so the short and the long robocolumns. And they're kind of adjustable to how many columns you are using on your deck, if you just use one strip of robocolumns or entire plate. So this is kind of adjustable. So yes, I'm super excited that we have or will have this option in this very near future, and we will definitely contact all our customers once we are ready.
All right. Jana, let's keep you in the hot seat for just another question. Somebody asked, I have several Hamiltons. Can I run the workflow on a Hamilton? Can you take that one?
Sure. Interesting question. Thank you. I'm not a Hamilton spokesperson, so I can only talk of what we do and maybe do differently from the Hamilton instruments. We run these robocolumns with a liquid displacement technology, and for this we have the liquid systems that we sell, and we need this liquid displacement because we build up a pressure of 2 to 3 bar in the columns and in the system. And this is only possible with a liquid system. And to my understanding, Hamilton does only have air displacement instruments. And for this, I assume that it will be difficult for them to run the columns. Yeah.
Okay, we're going to switch back to, we have quite a few questions about the software. So let's spend a few minutes there. Anonymous is asking, do we have a solution for having different residence times per column? I'm going to start that with Arne, but if any of the other speakers want to chime in there, we can certainly do that.
Yeah, so what we chose to, we support that. You can specify within the ANTA elements is actually get the graphical user interface, and you can specify residence time per individual column. Now, what we do in the software, the planning software, sort of groups all the same residence times together, and it will do all the elutions across those same residence times. And then it will go to the next, what we call strip of columns. Could be 2, 3, could be up to 8. So we group similar residence times together. They don't have to be adjacent, because we can control all 8 channels individually. But we then go and elute with similar sessions until all the steps in the elute series are completed, and then we go to the next flow rate, essentially. I hope that was clear.
So Arne, if somebody already has a TCAN workstation and they want to have an on-site demonstration, can they set that up through their website, or what's the best way for them to get a demo of the workflow?
Yeah, so we do lots of custom demo requests. That's not a problem at all. You can email us at demo@synthase.com. We can have generic demos, but we also allow you to give us your configuration using TCAN's export tooling. You can export actually the configuration of your Evo or even your Fluent quite easily. We can then import that, and then you will see your deck in that live custom demo. So that's something that we sometimes do as well. If you don't, we can go either way. We can go generic, or we can go customized to your specific deck layout or configuration. However many Roma arms you may have, or you might have an MCA in there, we can then tailor it to that.
So let's piggyback off of that. In addition to the demo, we have Hubert is asking, they have diverse devices attached to their TCAN, so plate reader, pH, conductivity modules from various suppliers. Does Antha integrate with those devices as well?
That will be on a device-by-device basis. We will integrate with some of them, but probably not all of them. However, we do supply the possibility to upload data from any device. We have this generic data upload capability. So for example, suppose that we don't support your specific pH meter that is on deck. If we have the data available in, like, let's say, a text file or a CSV file, that can be uploaded and it will be aligned with all the other data, and a report can be generated including those pH measurements. So that's what we call the first level of integration. We then typically, if many customers ask for the same integration, we then go to what we call a level 2 integration, where we sort of control that data acquisition as well. And so then everything is automated. Then you don't have to juggle manual files. So the answer really is it depends on the equipment. And then in any case, we provide the ability to upload data from that equipment through our generic data upload mechanism.
Arne, we have a couple of questions about the DOE and where it's designed. Can you touch on the program requirements for the DOE, and then just briefly comment if Antha can do the DOE itself, or if the DOE has to be imported from another program?
So that's a really, really good question. So within Antha, you actually don't need JMP or you actually don't need DOE design files. You could, in principle, specify your DOE within Antha's native user interface. That's for complex DOEs that becomes pretty difficult. So that's why we offer the ability to import JMP files or design expert files, any Excel file really. Like, it doesn't matter where it comes from as long as it's a tabular format. That's just, if you have complex DOEs, you don't have to sort of manually select it. But through its graphical user interface, you can select things like different flow rates, different buffers, different fractions that you want to collect. Also, that is natively supported through the user interface. We have a R&D project ongoing it's not released yet, but we are working on also designing your own DOEs. That's still in prototype phase, so that's not released, but that's something that we're at least thinking about in terms of designing your own DOEs.
Okay. Arne, can you provide a little more detail about how HPLC data would be incorporated into the system and then into the data analysis?
Yeah, it's a good question. So let's say you ran a RoboColumn workflow. You collect your samples in let's say deep well plates for sake of argument here. You can then essentially pick which fractions you want to select. You could potentially pool them from within Antha. So when you say, like, hey, I just want to send all my fractions to HPLC, or say I only want to send certain fractions with a certain concentration, like with a certain A280 above a certain value, that's also possible. That's possible from within Antha. So this is what we call a cherry pick workflow. So Antha will then generate instructions for the robot to go pick the samples, potentially pool them, and put them in another plate. That plate can then be sent to your HPLC team, or if you have some other foundry doing that for you, they will receive that plate, and typically what you get back is a CSV or an Excel file with sort of let's say monomer aggregates or fraction information. When you have that, Anta actually knows which samples these sets of information correspond with, because Anta has calculated all the liquid handling. It can also calculate or backtrace sort of these liquid handling steps. So you upload that data to the plate that was sent to the analytics team, and Anta will then backtrack all the liquid handling steps that happened, even if there was a dilution in there, and then tie that data back to the operational conditions of that experiment. So for each robocolumn and whatever the conditions were, you will see the values of, let's say, percentage monomer or aggregates or fractions.
Okay, let's talk a little bit about the learning curve of getting up to speed. So, let's say somebody is already using Opus RoboColumns and they have their TCAN workstation, but they want to integrate with Antha. Let's talk a little bit about how long that takes to get up and running. And also, Arna, if you could, can you do a little bit of compare contrast with the TCAN provided programming? And is there anything that the Antha software does to help overcome some of the programming hurdles that users may experience with that TCAN software?
Yes. So let's go to the first part of the question. Let's say you sign up for an Antha license. What will typically happen is you will get training, or we have a customer success team that are experts with the software, and this training will take typically 1 to 2 days. So after this 1 or 2 days, you will be accustomed to using the Antha user interface, and you will be able to generate instructions. There is a little bit of a variable part, as you know. These TCAN instruments, whether it's Evo or Fluent, can come in all sorts of sizes, and explained. So there is a bit of a variable aspect there that we always want to check that your configuration is fully supported within Antha. But that typically takes somewhere between 2 to 4 weeks. So if you put that aside, we have, let's say, the 2 days of what we call in silico training. That's just in the cloud. You don't even need your physical hardware present. And then we typically have a 2 to 4 week period before you can actually run on the device, and that depends on the complexity of the device configuration that you have. So in principle, within 2 to 4 weeks, you'll be up and running. That's what we aim for. And now to contrast that with TCAN, if you start from zero, typically you will get a training by TCAN, which takes probably 3 or 4 days. But the big contrast between Antha and the TCAN approach is that you can simulate, you can prepare your workflows in the cloud from anywhere. You don't need to be in front of a computer. Well, sorry, you need to be in front of a laptop, not in front of the lab computer. That's what I meant. So there's sort of, 2 or 3 users can do that simultaneously. It doesn't matter if 5, 10. It's like browsing. It's a browser application. You can then preview and you get a sense of what is going to happen. So in contrast, if you use, for example, the tChrome Wizard application, that application runs on the device connected to the to the robot. So on the computer connected to the device, essentially. And that's just a step, because then you sort of need to reserve the computer and you need to make sure nobody else is using the equipment. And yeah, you're in the lab, all with your protective equipment. It's less. It's not as easy to iterate across your design. Let's put it that way, compared to our solution. Yeah, I hope that answers the question.
I'll touch on this a little bit more, but there will be an opportunity for follow-up. We'll be sending out an email afterwards. So if anybody has additional questions, we'll make sure that they can get in touch with either you or somebody from Synthase. So no worries there. We're going to take one last question before we wrap here, from Marco, who commented, exciting case study. Was the best performance clustered in one area, or were there several areas that yield similarly good results? So Arna, I'm going to assume this is for the case study, and if you could just sort of briefly talk about where the best performance was.
Yeah, so that's a really good question, actually. The best performance was sort of clustered around a certain type of resin. And the reason for that is that I think the parameters were optimized sort of for inside that for that resin that resin operational conditions. So the more resins you do, sort of the more wide your design space becomes. What we noticed is that for some resins, actually, our pH range was not sufficient. So what we did is we actually did a follow-up study. We're analyzing those results actually now. So what you notice is like an area of optimized performance, but also an area of promise where we realized we didn't include the entire design space around that area of promise. So what we do is we're going to expand the design space around that area of promise and then see how these two regions of optimal performance will compare. Once we have those results.
Thanks, Arne. So I know I said that was the last question, but I'm actually going to take one more. Tim, can you comment for us? Sorry. The audience has submitted great questions today. I'm really excited to see all of this participation, so thank you. But one more for Tim here. Are there RoboColumns available off the shelf, or is everything custom and made to order?
Yeah, this is answer, because as you are aware, the diversity in available resins is almost including 300 different resins, commercially available. So current stages or current best practices that all RoboColumns are made to order exclusively for youth according to your demand. But we are currently looking at our historical data, and we are going to identify more or less frequently used resins, for example, for molecular antibody purification or for viral vector purification, which is getting more and more popular. And then we are considering to put those columns on stock and supply them with a very short lead time off the shelf.
Thanks, Tim. As I sort of mentioned earlier, Tim's going to be sending each of you an email shortly after we conclude the webinar to discuss next steps with anyone who's interested in establishing this integrated workflow in their lab. Before we wrap for today, I'm going to post a link in the Q&A panel to Repligen's event page. From there, you can access all of our previous webinar episodes, and you can also register for future events. At the end of June, we'll be hosting episode 4 of our 2021 webinar series. In episode 4, we'll be staying downstream and focusing on a new small-scale TFF system that's fully automated with modern software. So I hope to see all of you there. And with that, I want to once again thank our speakers, thank each of you in the audience for spending some time with us today. Enjoy the rest of your day and take care.